Okay, we're all set live. Sorry, that took a bit longer than usual. I had some hit the wrong buttons, but I think we are all set. And so we'll just wait a couple minutes. People usually pop in about a minute after the hour, and then we'll get started.
Sounds
and
good.
Ankita, you're all set. You're gonna lead today. Great.
I realized, Tina, you did your undergrad around this area. I'm at NIH right now.
Yep, I grew up in the DC area.
Oh, cool. Do you miss it?
Yes, but I don't miss the traffic.
True.
Yeah, I feel like in the Midwest, it's a whole different beast, right? You just never have to sit in traffic and you get really used to it. My sister lives in Missouri, so when I go visit her, I'm like, wow, you don't have to worry about going somewhere at 5 p.m. Like it doesn't occur to you not to go somewhere at 5 p.m. It's really wild.
Okay, do you think we should start maybe? People will trickle in while I'm doing the introduction.
Yeah, it's up to you. I always, you know me, I always start late. That's who I am.
No, I want to do justice to Tina's talk.
Fair enough.
Okay, welcome everyone. Thank you for joining us today again. Today we have Professor Tina Tootle. She received her undergrad degree in microbiology from University of Maryland, College Park, and her PhD in biology from MIT, working with Dr. Ilaria Ribe, where she studied RAS and MAPK signaling and the retinal determination pathway using Drosophila. Dr. Tootle did her postdoctoral study in the lab of Dr. Alan Spradling in Carnegie Institute of Science, Department of Embryology. It is there where she began studies on prostaglandin signaling and its role in regulating cell shape and migration during Drosophila oogenesis. She began her independent career at the University of Iowa in 2009 and received tenure in 2016. There she leads her research program focusing on prostaglandins, collective cell migration, lipid droplets, and nuclear actin. And today she's going to talk about Fascin and its role in collective cell migration and invasion. Thank you so much, Tina, for joining us today. And I would give you the stage. And really, we are very, very excited to hear your study and talk. Thank you.
Great. Thank you so much. I want to thank the organizers for both giving me the opportunity to give this talk, but also for starting this seminar series during a time when we really needed it. It has been wonderful both for my lab and the students at the University of Iowa. So first and foremost, today, I want to acknowledge that we're in a very difficult time and understand that science is not going on and life is not going on as normal. But I hope you can take this hour and enjoy some science along the way. So for this talk, please feel free to interrupt me with questions. To facilitate that, I'm going to take a pause halfway through to get questions, allow you to stretch or wake up if I've happened to put you to sleep, which I hope I don't. So this work that I'm going to tell you about is the work of one very talented PhD student, Maureen Lamb, who defended her thesis last year. She completed 2 first author publications in developmental dynamics in eLife, and she's currently a postdoctoral scholar in the lab of Paul Trainer at Stowers Institute. So in this group, I don't need to tell you this, but cell migration is an essential process. It allows tissues to develop and organs to form. Cell migration also plays critical roles in other processes in the body, including tissue repair and wound healing, such as our epidermis. Cell migration is also hijacked to mediate cancer metastasis. And while we originally thought many cancers migrated only as single cells out of the tumor, there's a growing body of literature really indicating that cancer cells can migrate as clusters or groups or migrate collectively. And these collectively migrating cancer cells may be more resistant to chemotherapies and more likely to seed metastases. Indeed, if we look at a spheroid of cancer growing in vitro, we can see this invasive collective front coming off of this cancer tumor. One contributing factor to regulating migration and invasion is substrate stiffness, or the stiffness of the environment. Data really suggests that increasing stiffness is promoting migration and invasion. And I want to just take a moment to talk about some of that data. So we know that with increasing stiffness and increasing confinement, that together both of these attributes can drive increased cell migration. So this red line here is indicating the most confined cells, and you can see this nice increase in cell migration speed with increasing stiffness of the environment. However, I want to point out that depending upon how confined these cells are, they reach a stiffness point in which their migration speed slows down. And this and other work, including the one I'm showing you here, indicates that specific cell types may have an ideal substrate stiffness for their fastest migration. So these are breast epithelial, either normal, non-invasive cancer or invasive cancer cell lines. And we can see the normal cell lines don't really care what stiffness of substrate they're on. They migrate at about the same rate. But the cancerous cells tend to prefer the higher stiffness to migrate faster. However, we see the most invasive cancer cell is migrating the fastest on intermediate stiffnesses. So this really suggests that the stiffness of the environment needs to be tuned to the particular cell that's migrating. And it may also mean that the fastest migration may not be ideal for a given circumstance. For example, during development, cell migrations need to be coordinated with other events that are happening, and therefore a cell needs to move at the speed best for that tissue or organ development. So as we think about how stiffness is driving migration, what it's doing is inducing a mechanotransduction cascade that ultimately changes the actin cytoskeleton to allow for cellular protrusions and other changes that mediate migration and invasion. And it's this actin remodeling I want to focus in on today. So when we think about a migrating cell, the actin cytoskeleton is highly dynamic. It sends out protrusions at the leading edge. These protrusions are anchored in adhesions that are providing the force for these cells to migrate. The cytoskeleton organizes and protects the nucleus and nuclear position. And ultimately, the trailing edge of the cell needs to contract and retract for the cell to be able to move forward. The way that actin is regulated is that there are many actin binding proteins. So actin exists as monomers that polymerize into these filaments. Filamentous actin or F-actin is regulated by things like exchange factors, nucleating factors, and elongation factors. Filament length is regulated by capping and severing proteins. And these actin filaments are then organized into structures such as branches and bundles. And these structures of actin are then organized into networks to allow cellular protrusions and other cellular structures. Today, I want to talk to you about one actin bundling protein called Fascin. Fascin is a monomeric actin bundling protein, and therefore it's the tightest known actin bundler. This means Fascin holds actin filaments very close together, giving them more rigidity. And this is why Fascin is so important for driving cellular extensions such as filopodia and invadopodia. Fascin promotes cell migration during many contexts. It regulates development, such as neuronal growth cone formation, but it also plays critical roles in cancer. Fascin is very weakly expressed in normal epithelial cells, but becomes massively upregulated during tumorigenesis. And it also is known to be a biomarker of aggressive cancers and poor patient outcomes due to its roles in driving migration and invasion. Much of what we know about Fascin's roles in cell migration have come from studying how it regulates migration in 2 dimensions, either as single cells or collectives. However, we all know that in the body, most cell migrations are occurring in 3 dimensions, and many of these are as collective cells or groups of cells migrating together to drive tissue or organ development and even cancer metastasis. Indeed, this brown color is Fascin being expressed in these migrating clusters of cancer cells. So one of the questions my lab is really interested in addressing is what are the roles of Fascin in invasive collective cell migration? And to do this, we take advantage of the robust genetic system of Drosophila and a particular migratory event during Drosophila oogenesis. You might wonder why bother to use a fruit fly when you could study this in a cell. So I want to take a moment to tell you why fruit flies are a great model system for cell biology and other areas of research. So flies have less genetic redundancy. you and I have 3 different Fascin proteins within our body. Flies have only 1 Fascin. We have large numbers of mutants available, including multiple null alleles for Fascin. Flies are really fast. You go from your cross to your next generation in 10 days. This means we can combine all of these variety of genetic mutants and other tools to perturb and regulate gene function. The best reasons for flies are the genetic tools. They allow us to misexpress or downregulate genes in particular cell types and at particular times during development. And while flies are a simple organism, they are sufficiently complex. They have analogous tissues and organs to that which we see in higher animals. And the processes, such as those controlling collective migration, are conserved from flies to higher eukaryotes. Specifically for studying Fascin in collective cell migration, we use a migration during Drosophila oogenesis. But for the purposes of this talk, you don't need to know anything about oogenesis. All you need to understand is the composition of a stage 9 follicle or developing egg. So I just want to take you through the cell types within this follicle. We have a posterior oocyte in gray, 15 anterior nurse cells in blue. These are covered in a somatic epithelium of follicle cells in purple. And the cells we're going to be focusing in on today are in orange. These are the border cells. These border cells delaminated from that purple epithelium and are migrating as a collective down between and on the nurse cells. So the nurse cells are the substrate for the border cell migration. Just to walk you through the process of border cell migration, I'll take you through this schematic. At the beginning of stage 9, the border cells are specified and they're in green, and they're going to delaminate and migrate between these much larger nurse cells until they reach their ultimate destination. Importantly for us, these border cells are in line with the position of the outer follicle cells shown here in gray. This allows us to determine at any point in the migration if border cell migration is on time, delayed, or accelerated. Now, I've told you that this is a collective and invasive cell migration, but I want to illustrate that. Sorry, I'm trying to make that. I'm just going to go back. Sorry about that. So this is a confocal stack, and we're looking at the surface of the follicle. These are those somatic follicle cells and the much larger underlying nurse cells. As we go down through the confocal stack, we get to the middle of the follicle, and you can see the border cell cluster pointed out by the yellow arrow. And it's surrounded on all sides by the nurse cells. And as I just showed you, above it are also the nurse cells. And if we continue down through the stack, you see there are nurse cells on all sides of the border cell cluster. Thus, this is an invasive and collective cell migration. So we can use this system to study the role of Fascin in this process, because Fascin is highly upregulated in these border cells. But I want to point out that Fascin is also expressed in the substrate, the nurse cells. So we can use this system to determine what are the roles of Fascin in invasive and collective cell migration. So here I'm showing you 2 wild type follicles. You can see the border cell cluster indicated by the yellow arrow. And the yellow line marks the position of the outer follicle cells. And in our wild type follicles, you can see that the border cell migration is on time as it's in line with the outer follicle cells. However, when we have Fascin null mutations, 2 different ones, we find that our border cells are significantly delayed compared to the position of the outer follicle cells, indicating Fascin is required for on-time migration. We want to quantify these defects, and to do this, we take advantage of the fact that the border cells are in line with the outer follicle cells. So we measure the distance the border cells are from the anterior end of the follicle and the distance the outer follicle cells are, and we do a simple division. This gives us our migration index. And if the border cells are on time, this ratio will be 1. And if they're delayed, they will be less than 1. So we see in our wild type and our Fascin heterozygotes that our average migration index is 1, indicating on-time migration, and that it's significantly less than 1 in our Fascin mutants, indicating delayed migration. So we have found that Fascin is acting in both the Fascin is required for border cell migration, and now we want to determine where Fascin is required. And remember, it's expressed in both the border cells and their substrate. So to do this, we take advantage of the UAS Gal4 system. In this system, we have a genomic enhancer, which is driving the expression of the transcription factor Gal4. Gal4 will bind to the upstream activating or sequence or UAS to drive whatever we placed downstream of this. In this case, an RNAi to Fascin. And we can take advantage of numerous Gal4 lines to knock down Fascin expression in the different cell types. Specifically, we're going to knock it down in all of the somatic cells, which are the follicle cells and border cells, in the border cells only, and in the substrate. So when we do this, we find that when we knock down Fascin in all of the follicle cells, including the border cells, or the border cells only, we have delayed migration. We also find that when we knock down Fascin in the substrate, we also see delayed migration, indicating Fascin is acting in both the migrating cells and their substrate to promote migration. So having found that by RNAi that Fascin functions in both the border cells and the substrates, we next wanted to test this using cell-specific Fascin rescue. Again, we turn to the UAS Gal4 system. In this case, we're going to express GFP Fascin in our Fascin mutants in these different cellular contexts to determine where Fascin is required. And we find that Fascin mutants have delayed migration, as I showed you before. And if we re-express Fascin in all of the follicle cells and the border cells, or the border cells only, we restore on-time migration. However, when we re-express Fascin in the substrate, we fail to restore on-time migration. This data indicates that Fascin is sufficient in the border cells for on-time migration and is not sufficient in the substrate. But we were a little surprised about the differences in our knockdown versus our rescue experiments. And we wondered if this could be due to the level of expression. So remember, Fascin is highly expressed in the border cells and very weakly expressed in the substrate. However, when we use the substrate Gal4 to express Fascin, we have a much higher level of Fascin in these substrate cells. And if we look at this, we can see that by overexpressing Fascin in the substrate, it results in a very abnormally shaped border cell cluster, suggesting that Fascin expression must be tightly regulated in the substrate for normal migration. So we wanted to more faithfully recapitulate the endogenous level of Fascin expression. So we used the UAS Gal4 system and a global Gal4. And this global Gal4 expresses strongly in the border cells and other somatic cells, and very weakly in the substrate, the nurse cells. And when we do this, we find that the delay in migration due to a loss of Fascin is rescued by expressing Fascin globally. So we have found that Fascin is acting in both the border cells and the substrate to promote migration. We next wanted to determine how Fascin is regulating border cell migration, so we turned to live imaging, expressing a membrane GFP within the border cells. And when we did this, what we observed is that Fascin plays a role in delamination. So the wild type cluster will round up and separate from the follicular epithelium in about 90 to 100 minutes. Whereas in our Fascin mutants, while the cluster rounds up and is trying to get off of the follicular epithelium, it takes much longer, often upwards of 300 minutes to come off of the follicle cell layer. Once we got border cells that are delaminated, we then looked at what was happening during migration. In our wild type border cell cluster, they generally have one main protrusion off the front of the cluster. And in our Fascin mutants, what I hope you can appreciate is that they extend extensions off of the cluster in all directions. This is perhaps easiest to see if we look at the stills of the live imaging. Here you can see the nice protrusions off the front of the cluster, and there are very rarely protrusions off the side or the back of the cluster. In our Fascin mutants, however, we see an increased number of protrusions, and these protrusions are localized to all sides of the cluster, not just at the leading edge. So ultimately, what we find is that in our Fascin mutants, there's a delayed migration speed compared to our wild type cluster. So far what I've shown you is that Fascin is required in both the migratory cells and their substrate for on-time migration. This loss of Fascin is causing delayed delamination or the initial detachment from the follicular epithelium and ultimately slower migration. Contributing to these defects are increased and mislocalized protrusions off the cluster. And in data I'm not telling you about today, we know that part of this is due to the misregulation of the actin elongation factor enabled, which is shown here as the purple stars. So enabled is the sole enavasp actin elongation factor member in Drosophila. It localizes to the tips of actin filaments and promotes their elongation. And it's known from in vitro studies that Fascin bundled actin filaments increase the processivity of enabled, meaning it stays on the filaments longer, driving increased polymerization and therefore longer protrusions. In our Fascin mutants, we're going to have a loss of that processivity, which will result in shorter protrusions that are less stable. And we know that overexpression of enabled can suppress the defects in our Fascin mutants. In other data I'm not telling you about, we also know that Fascin is playing a role in regulating E-cadherin-based cellular adhesions, where we have this upregulation of E-cadherin at the border cell substrate boundary and also between the substrate nurse cells themselves. Additionally, there are likely other roles of Fascin in this migration, one of which I'll tell you about in a little bit. But I first want to pause and emphasize why it's so important that we found that Fascin is acting not only in the migrating cells, the border cells in orange, but their substrate, the nurse cells.
Is this your pause, or are you having a pause in a minute?
In one slide.
Okay.
And I just want to emphasize that in the literature, we focused on the role of Fascin cell autonomously within the migrating cells because of its critical role in driving filopodia and invatopodia formation. And I think our data on border cell migration really suggests we need to think about what Fascin is doing in the environment of these migrating cells, even when Fascin is only weakly expressed. And so now we are to that break. So Claire, if you have a question, I'd love to hear it.
I have two questions. One is, it blows my mind that upregulation in the invasive cells. I mean, is this like an EMT marker? And have people looked at whether in tumor cells it getting rid of it prevents invasion? That's my first question.
Oh yes, Fascin has been well studied in tumor biology. It plays a critical role in invasion and promotes it. It's not absolutely essential. You slow invasion when you perturb Fascin. You don't block it completely.
Okay, so the second question is, it makes no sense to me that getting rid of Fascin and reducing the activity of enavasp would increase protrusions. That's completely counterintuitive.
Right, so it's not that it's getting rid of enavasp, it's that we're no longer targeting enavasp to be processive at a particular location. So if Fascin
so you find that the enavasp is mislocalized to these errant protrusions?
So it is hard to tell from the imaging if there is a change in enrichment. The enavasp antibody is sort of a pill. But what we think is that because there are, and it's probably there are other reasons why there are protrusions at these different locations, but we're not targeting enavasp activity and maintaining it at the right space to drive successful protrusions. So we see more protrusions, but these protrusions retract much faster, so they're not successful.
Thank you.
Great. Are there other questions?
Can I ask?
Please go ahead.
Can you clarify, maybe it's semantic, but like border cells are actually squeezing through cells or sort of like invading, like we would think an invasion into matrices?
They're moving between cells. And I forgot to mention that there's little evidence of extracellular matrix on the outside of these border cells, although I think there is a low level that's being secreted by these cells themselves. So it's really a cell on cell migration, and it's pushing between the cells and not breaking the cells.
Thank you.
I think we have a question in the chat as well from Curtis. For Fascin knockdown, is there a difference in the speed or directionality of the border cell cluster post delamination? How does the phenotype compare with the RAC knockout cells?
Yeah, so there is a decrease in the migration when we look at ones that have delaminated and are in the migration path. We see about half the rate of migration speed during sort of the mid-migration of the border cells. The RAC knockout really is it is somewhat similar in that it clearly doesn't have a polarization. I would say Fascin mutants probably ultimately migrate better than a RAC knockout because you absolutely need that RAC to move.
Thank you.
Great. We have another question from Pedro Lowenstein asking, is Fascin expressed in the area surrounding tumor cells?
Yeah, I think that's a great question, and I don't think anyone has asked. I will say that all of the Cancer Genome Atlas data, which took tumors and likely surrounding cells to them, there is high upregulation of Fascin in those aggressive samples with poor patient outcomes. But I don't really think we know what Fascin is doing in the microenvironment of these tumors, and I hope some of you who study that will be interested in looking at it.
Thank you. I think Sandrine has a question. Sandrine, if you would like to go ahead. I think you're muted.
Okay, now
There
okay.
you
go. Sorry. Yes, I had a question about the link between Fascin and the cadherin expression. So the first question was whether the change in expression in cadherin is also observed in the nurse cells, where you said that Fascin was also playing a role, and what could be the importance of this in the fact that the cells have trouble migrating?
Yeah, so we do see increased E-cadherin between the nurse cells themselves. We really haven't figured out what this is doing, but it may relate to the second half of the talk. So we haven't pursued what Fascin is doing with regulating E-cadherin, but we're definitely interested in understanding it.
Thank you.
Great. I think this intermission was a really good idea because we have a lot of questions. Here's one from Natalie Hussein. In the Fascin mutant border cells, they're able to delaminate, cluster, and migrate. So what could be the compensatory proteins triggering the collective behavior?
Yes, so Fascin is not going to be the only bundler that's present in these cells. It's also not the only way to send a cellular extension. So I imagine that there's enough enabled activity that they're somehow moving and sending enough protrusions to move. And we do know that when we overexpress enabled, we can suppress many of these defects, although we didn't specifically look at delamination. We do decrease the delay in migration when we overexpress Fascin. I overexpress ENA in the Fascin mutants.
All right, great. I actually have one quick question, if you don't mind, before you move on. So I did not know that the migration of the border cells was coordinated with the migration of the outer, the follicle cells. So is that a sort of a coordinated, actively maintained process that they're migrating in sync, or did they just, under normal circumstances, kind of happen to move at the same speed, but they're not at all actively coordinated?
Right. So the outer follicle cells aren't technically migrating. It's that the squamous cells in front of them are forming and sort of the whole follicle is growing. So that's a semantic definition. I don't know why they're in sync. But I do think it is very intriguing. Ultimately, they're going to get to the same position that ultimately the border cells migrate up to. So there's something about their connection that we don't understand.
That's so fascinating. If they've just evolved to normally go together, or if there's some maintenance of that causes them to move together.
Yeah,
It's
there are definitely mutants that have outer follicle cell problems, and I have not gone to look and see how that impacts, or if it impacts, the border cells.
Yeah, that's so interesting. Okay, I think we've got everyone for now, so please continue. Thanks so much.
Great, thank you. All right, so I've shown you that Fascin is required in both the border cell cluster and the substrate. And this finding that Fascin has a role in the microenvironment of these migrating cells really intrigued us. And it made us wonder what it might be doing there. And we thought perhaps it's regulating tissue stiffness. So as I mentioned at the beginning, there's a lot of evidence that increasing the stiffness of the environment can drive mechanotransduction, to drive actin remodeling, and ultimately migration and invasion. And this is well known that this is happening within the border cells. So Jocelyn McDonald's lab has nicely shown that stiffness of the substrate, the NER cells, does impact the border cells. These migrating border cells are then going to respond and give a reciprocal force back. And this balance of forces between the microenvironment, the substrate, and the border cells is essential for on-time migration. And I just want to emphasize, just like many other contexts, the border cell force balance seems to be primarily regulated from the substrate based on the literature. And so the cellular means of controlling tissue and cellular stiffness is non-muscle myosin, which I'll refer to as myosin. So myosin is composed of two heavy chains, which allow the dimerization and also have the head domain, which binds to actin and has ATPase activity. Then we have two essential light chains and two regulatory light chains. And myosin activity is regulated by phosphorylation on that regulatory light chain, such that phosphorylated myosin is active. This active myosin is going to form bipolar filaments, allowing it to bind to different actin filaments and allowing to contract them. This contraction ultimately will give cortical tension to the cortical actin and therefore cellular stiffness. Importantly, myosin is required for on-time border cell migration. Loss of myosin results in failures of delamination and altered protrusions off all sides of the cluster. And I just want to remind you, these are the same defects we saw in our Fascin mutant. And when you see similar defects, it suggests that maybe these two factors function in the same pathway. And indeed, there was an in vitro study that implicated Fascin in regulating myosin. Specifically, Fascin bundled actin filaments limit myosin activity. And the way they do this is because Fascin, or thought to do this, is because Fascin bundles actin so tightly, it's preventing myosin from being able to access these filaments and bind to them. And that's how it's inhibiting myosin.
Just a real quick question there. It's not
Yes.
having any effect. There's no feedback on the phosphorylation of light chain in that case, or it's really a steric thing.
So there was no evidence that it was regulating the phosphorylation.
Thank you.
But it's only one study. So these data led us to hypothesize that Fascin is limiting myosin to control stiffness to ultimately regulate border cell migration. And so we first wanted to look at whether Fascin is indeed affecting myosin activity within the border cell cluster. And we're going to look at this by looking at the phosphorylation of the myosin regulatory light chain. So as has been published before, there is increased active myosin on the surface of the border cell cluster. And when we get rid of Fascin, we see a striking increase in the level of active myosin, not only on the border cell cluster, but also on the NER cell or substrate membranes. And if we zoom in on the border cell cluster and color this based on intensity, I hope you can really appreciate how much more active myosin is on the surface of these border cells. So we wanted to quantify these changes using relative fluorescence intensity. And so we first looked at the substrate, And we see that indeed in our Fascin mutants, we have increased levels of active myosin on the membranes of our nurse cell substrate. We also see increased active myosin on the surface of our border cells. When we looked at this myosin activity on the border cells more closely, we noticed that there are increased number of active myosin puncta per border cell cluster, but these puncta length are shorter. And this suggested to us that the localization of myosin activity and the distribution or dynamics of it, its on and off cycling, are impacted by the loss of Fascin. So we wanted to look at this more closely, and we turned to live imaging using a GFP tagged myosin regulatory light chain. And it's thought that these little flashes of puncta on the surface of the border cells are indicative of active myosin. If we compare that to what we see in our Fascin mutants, what I hope you can appreciate is that the active myosin is moving much more slowly around the surface of the border cell cluster. And we quantify this, we can see indeed that the puncta lifetime is about twofold increased compared to our control for the myosin regulatory light chain. So these data support that in vivo Fascin can indeed limit myosin activity, not only in the migrating cells, but their substrate, the nurse cells. So we next wanted to address whether this has any impact on tissue stiffness. So while myosin activity is a nice readout or proxy of tissue stiffness, we wanted to get at this more directly. And so we turned to atomic force microscopy. So in atomic force microscopy, you have a cantilever, and this can be thought of as a spring. It's going to be lowered down into our sample, the stage 9 follicle, and based on how stiff or soft this substrate, this sample is, it will bend the cantilever, and that bend will be detected by a laser beam. So if we have a stiff sample, it will bend the cantilever less, and if we have a soft sample, a stiff sample will bend the cantilever more, and a soft sample will bend the cantilever less. So we are not atomic force microscopists, so we collaborated with the Tavansky lab in the chemistry department at the University of Iowa and his very talented graduate students. So when we do atomic force microscopy on the Drosophila follicle, we need to think of the layers of the follicle. So on the outside of the follicle is a basement membrane shown in gray. Underlying this are those somatic follicle cells, both the stretch cells in yellow and the outer follicle cells in purple. And then we get to our substrate in blue, the nurse cells. So in order to understand the stiffness of the nurse cells, we need to think about separating our data from our atomic force microscopy studies, such that we look at the data from 0 to 100 nanometers to measure our basement membrane stiffness, and we look at the data at 310 to 550 nanometers to get at the nurse cell stiffness. So let's first look at the basement membrane stiffness. So if we indent from 0 to 110 nanometers, we see that in both wild type and our Fascin mutants, we have a stiffness around 25 kilopascals. And this is comparable to what others have seen doing similar studies. If we then look at the stiffness in the 310 to 550 nanometers, which will be our nurse cells, we see that our wild type nurse cells are softer than our basement membrane, and this has been published before by others. And we find that in our Fascin mutants, the nurse cells are not softer than the basement membrane and are 2 times stiffer than our wild type, indicating Fascin is indeed controlling substrate stiffness. So we found that Fascin is limiting myosin activity, and this is ultimately affecting the stiffness of the nurse cells. But does this have anything to do with border cell migration? So to begin to address this, we first turn to pharmacology using the Y27632 Rho kinase inhibitor. So Rho kinase phosphorylates myosin. So if we inhibit this kinase, we will decrease myosin activity. Our Fascin mutants, as I've shown you before, have delayed migration. If we treat with the Rho kinase inhibitor, we restore migration. We also took a genetic approach to address this, knocking down the myosin regulatory light chain, specifically in the substrate. Again, our Fascin mutants have delayed migration, but when we decrease myosin activity in the substrate, we can restore on-time migration. So together, this data leads to the model that Fascin is limiting myosin activity to control substrate stiffness, and this plays a role in promoting border cell migration. So we next wanted to get at how is Fascin regulating myosin? So the in vitro study implicated that it is Fascin bundling of actin filaments that's somehow limiting myosin activity. And luckily for us, we know that if we phosphorylate Fascin at serine 52 in Drosophila or serine 39 in higher eukaryotes, that this phosphorylated Fascin no longer bundles actin filaments. And this would allow myosin to come in and bind to those filaments and be active. So we asked whether expression of a phosphomimetic Fascin will fail to rescue the myosin activity in the Fascin mutants using our UAS-GAL system to express a phosphomimetic form of Fascin globally, which means it's strongly expressed in the border cells and weakly expressed in the substrate, just like wild-type Fascin is. And when we do this, our Fascin mutants, as we've shown you before, have increased active myosin on both the border cells shown by the orange arrowheads and on the nurse cell membrane shown by the blue arrowheads. If we re-express Fascin using the global GAL4, we suppress this increase in active myosin. However, if we express the phosphomimetic form of Fascin, we see we retain high levels of active myosin on both the border cells and the substrate membranes. So these data indicate the actin bundling activity of Fascin is required to limit myosin activity. We then asked where in the follicle does Fascin regulate myosin activity? And we asked this because remember, Fascin is expressed in both the border cells and the substrate, and its activities in both of those play a role in border cell migration. So we again turn to our UAS-GAL4 system to knock down Fascin in these different cell types and determine what happens. And I just want to go through what we expected based on the prior literature. So the prior literature indicated that the nurse cells or the substrate is driving this force balance with the migrating cells to promote migration. Therefore, we expected that Fascin knockdown in the substrate would increase myosin activation in both the substrate and the border cell cluster. We also expected that Fascin knockdown in the border cells would increase myosin activity in the border cell cluster, but this would have little to no effect on the substrate. So when we knock down Fascin in the substrate, we indeed find increased active myosin on those substrate cells. However, unexpectedly, we find that when we knock down Fascin in the substrate, we fail to increase active myosin on the border cell cluster. Conversely, if we knock down Fascin in the follicle cells and border cells, or the border cells only, we see this increase in active myosin on the border cells as we expected. However, surprisingly, we found that if we knock down Fascin in the follicle cells and border cells, or border cells alone, we increase active myosin on the substrate cells, indicating Fascin can act in the border cells to control myosin activity in the substrate. We then went on to look at whether this alters substrate stiffness using atomic force spectroscopy, and knocking down Fascin in the substrate does indeed increase substrate stiffness. However, knocking down Fascin in the border cells only is sufficient to increase substrate stiffness. So this leads to the idea that Fascin is likely acting in the border cells to control substrate stiffness. But we wanted to address this a little bit more because we were surprised by it and wanted to know if Fascin activity in the border cells was sufficient to control substrate stiffness. And to do this, we used our UAS-GAL4 system to re-express Fascin in the border cells only of our Fascin null mutants. So when we do this, our Fascin null mutants have increased active myosin on the border cell cluster, and this is suppressed by re-expressing Fascin in the border cells. Similarly, Fascin mutants have high levels of active myosin in their substrate, and when we re-express Fascin only in the border cells, we can restore normal substrate myosin activity. We also see that we restore normal substrate stiffness when we re-express Fascin only in the border cells. So we had expected that the nurse cells were the primary driver of this force balance between the border cells and the nurse cells, the substrate on which the border cells migrate. However, what we found is that in the case of Fascin activity in the border cells is driving this force balance, such that it's limiting myosin activity in the border cells to ultimately control the stiffness of the surrounding substrate. And that promotes migration. So this was a surprising finding, and we wanted to see if it was a Fascin-specific phenomenon. So we decided to express catalytically active Rho kinase, constitutively active Rho kinase in the follicle cells and border cells. So Rho kinase will phosphorylate myosin and increase myosin activity. And we find that when we express constitutively active Rho kinase in the border cells, we increase activated myosin on the border cells as expected. And we also find that this is sufficient to increase activated myosin on the nerve cell substrate. So what I've shown you today is Fascin is playing a critical role in the border cells to control substrate stiffness and therefore migration. So how we envision this is working is that Fascin bundled actin filaments, which in a wild type nerve cell should be largely at the leading edge, is locally inhibiting myosin activity. And this myosin activity then is propagated through the border cell cluster to set the right level of border cell stiffness. This stiffness is sensed by the surrounding nerve cell substrate, which respond by appropriately regulating myosin activity and cellular stiffness. It is important to point out that Fascin in the substrate does have a role in regulating substrate stiffness, but this is not sufficient to control border cell stiffness or border cell migration. In our Fascin mutants, what we find is that we have increased and altered dynamics of myosin activity within the border cell cluster. This results in likely a stiffer border cell. This is sensed by the surrounding substrate, which correspondingly upregulates and activates myosin to stiffen the microenvironment. And this impairs the migration. So this finding that Fascin is acting within the migrating cells to control substrate stiffness seems at odds with a lot of the literature where we see that altering the stiffness of the substrate drives cell migration. But I don't think it is, because when we think about the context in which we've looked at substrate stiffness driving migration, we've used polyacrylamide gels with ECM on them. And these polyacrylamide gels with ECM are not able to propagate a stiffness change throughout the whole environment. They can't respond biochemically to what's happening. And indeed, we do know that even in these environments, these migrating cells can modulate the local environment. So here we see two different migrating cell types, and the background has been color coded for low stiffness in yellow and higher stiffness in reds and orange. And we can see that at the leading edge of these cells, that these migrating cells are indeed making the environment around their leading edge stiffer. It's also important to think about the differences between single and collective cell migration. Collectively migrating cells are known to be better able to sense stiffness changes, and they also place 4 times more force on the substrate. Additionally, it comes to mind as whether this process of migrating cells controlling their environment is occurring in other contexts besides border cell migration. It is well known that cells like to migrate on a gradient of stiffness. This is called durotaxis, so moving towards the stiffer environment. And this is seen in limb budding, neural crest development, cancer metastasis, and wound healing and fibrosis. And I want to raise the question of, is the environment driving the migration of these cells? Is that stiffness gradient already there? Or are the migrating cells either initiating or regulating that stiffness gradient to promote their own migration? And last, I want to bring up the idea of whether this change in tissue stiffness has any role beyond promoting cell migration. These border cells are migrating on these nerve cells, and they're changing the stiffness of these nerve cells during the process of migration. Is that stiffness a permanent change in migration? Does the stiffness have a role in setting up those cells for later developmental events? This is particularly intriguing given these cells are going to significantly strengthen their cortical actin and become contractile a few stages later in oogenesis. And I like to think that perhaps this isn't a border cell specific phenomenon, but is happening widely in development. Primordial germ cells undergo an extensive migration during embryogenesis, often migrating through tissues, including the gut. Does this migration through these tissues change the tissues they're moving through? Does that have a role in later events within this tissue development? And indeed, we've seen from the Seekhouse lab that the macrophages in Drosophila are also migrating through tissues, and seemingly they might be changing their environment. So with that, I'd like to summarize what I've shown you today. Fascin is required in the border cells and the substrate to promote on-time border cell migration. It's controlling the initial invasion or delamination of these cells. It regulates the directionality of protrusions and ultimately migration speed, and it's non-cell autonomously regulating the stiffness of the substrate. And we think this is not a fly-specific phenomenon, but Fascin is probably playing these other roles in its context of developmental cell migrations and perhaps cancer metastasis. And while I've told you the role of Fascin in a bundling-centric mechanism, Fascin is much more than an actin bundling protein. So today I've talked about Fascin tightly bundling actin filaments to both promote the processivity of the actin elongation factor enabled, but also to inhibit non-muscle myosin 2. However, we and others have shown that Fascin have many other roles in the cell. Fascin can cross-link actin and microtubules. This may play a role in cytoskeletal dynamics during collective cell migrations. Fascin also acts as an adapter between actin and the link complex. The link complex is the linker of the nucleoskeletal and cytoskeletal complex. This allows forced transmission from the cytoskeleton into the nucleus, allowing the nucleus to respond to these forces, perhaps by softening to allow for the cells to migrate through a tight space. We also know that Fascin localizes to the nucleus, and while we don't understand what Fascin's doing there, we have implicated it in regulating nuclear actin and the nucleolus. So we think that all of these diverse functions of Fascin likely contribute to its roles in border cell migration, but also its roles in other migratory contexts. And we think that border cell migration is a great in vivo model to really begin to dissect how all of these different functions of Fascin contribute to invasive collective cell migration, where these different functions of Fascin are important in the migratory cells versus their substrate, and how are these different functions of Fascin coordinately regulated. And as you may know, we think one of the means of coordinating the roles of Fascin is through lipid signals called prostaglandins. So with that, I want to acknowledge my lab. Again, this was the work of one graduate student, Maureen Lamb, who's now a postdoc. The border cell studies are continued by Sam and Ashley in the lab. I want to acknowledge our collaborator, Alexei Tavinsky, sources of reagents and funding, the great fly community at the University of Iowa. And I also want to make a selfish plug. We have a new MIRA that's about to be funded, and I would love to recruit a postdoc to continue these studies. So with that, I thank you for your attention, and I'm happy to take any questions.
Thank you so much, Tina, for such a fantastic talk. I would like to use my privileges to first ask the question. In your model where you have a Fascin knockdown, there is an upregulation of phosphorylated myosin, and that sort of is not making sense in my head. How would that lead to more protrusions everywhere? Like I would think that it would just, keep all those protrusions. And this is also linking what Claire was asking initially.
Yeah, so I don't think, it's hard to know if these cells are normally generating and sensing a stiffness gradient. And if now we've altered where the stiffness is occurring, and so there's trying to find the stiffness gradient by sending all these protrusions. So I agree, everything is stiffer, but I don't think it's so stiff that it can't send a cellular protrusion.
Okay, thank you. Go ahead, Jen, sorry.
Yeah, great, thanks. So we do have a lot of questions. Folks, as always, feel free to raise your hand or type question in the chat. We'll do our best to sort of ask them in the order they were given. So two questions from Harun Qureshi. First, asking about for your fluorescently tagged myosin regulatory light chain, that since it won't tell whether or not it's phosphorylated. How do you sort of justify talking about live imaging of the myosin regulatory light chain as the active myosin dynamics?
Yeah, so what I will say is that usually when myosin is not active, it's more diffuse. And so it's those puncta that we're really focusing on. So where you have an enrichment of this myosin regulatory light chain has been used as a proxy for active myosin. I agree it's not perfect, but it does correlate with what we're seeing with our fixed staining. There are some more recent biosensors for myosin activity that might be able to be used as well.
Okay, and this might have been partially answered, but is also asking if you address if Fascin is regulating myosin phosphatase activity or somehow sequestering its activity in the bundles, thus limiting the myosin phosphorylation.
So we have not addressed at all if Fascin is regulating the myosin phosphatase. I think there are probably roles of Fascin in inhibiting myosin activity beyond just the bundling of actin. They're just challenging to get at.
Great, thanks so much.
Thank you. Next question is from Ernest Liu. Do you have an actin binding deficient mutant of Fascin? It would clarify the role of Fascin in regulating myosin. So it's more of a comment.
Right. So what I would say is that the phosphorylation of myosin at the serine 59 not only blocks bundling, but it's sufficient. It in vitro studies decreases binding to actin filaments. It's challenging. There are some other mutants that may affect actin binding, but what I want to emphasize is it doesn't really get at which actin it is, because remember, it's probably connecting to the link complex, and that requires dynamic changes in phosphorylation and retention of certain actin binding domains. So there are 3 actin binding domains on Fascin, and on one side they act cooperatively.
Thank you.
Okay, great. We have a question from Nile Taneja asking about the effect of the Y compound on migration speed of the border cells in the wild type flies.
Right. So I don't remember our data, but obviously people have previously used this compound to inhibit myosin activation, and that would perturb the migration of the border cells on its own. So it's really the fact that in our Fascin mutants, we're actually restoring and making on-time migration with a drug that would normally impair it.
Thank you. The next question is from Tae-Hugh Kim. During the migration, how are the migrating cells attaching to the substrate cells, and does Fascin regulate those contact points? I think this is going to the E-cadherin.
Yeah, so there's definitely some idea that E-cadherin is providing that migratory attachment. We also have some data, although it's not widely appreciated in the border cell field, that maybe there's some role of integrins in ECM. There's very little ECM around these border cells, if any. We are starting to see some ECM. We know that in Fascin mutants, integrin localization to the surface of the cluster is perturbed. So I also think there's probably some integrin role in there. So I don't know if they're stuck more with E-cadherin and providing less force with integrin, but that's sort of what I think is going on at the moment, but we don't know.
Thank you. I think next was Sandrine, actually, who raised her hand. Sandrine, if you would want to go.
Hi again. Very interesting talk. Had a question about the dynamics of these changes in the mechanics of the cells, meaning in the nurse cells. You say that you have an increase in the stiffness as the cells migrate through, but how long does it last? So have you checked this, the mechanics of these cells before and after migration?
Yeah, so we haven't. So what I will say, I mean, that's an area we're really interested in. There is data from other labs that as the follicles progress, generally follicle stiffness is increasing with follicle development. So what I would say is I would predict and don't have explicit data that an early 9 stage 9 follicle is softer than a late stage 9 follicle. We tried to only look at mid-migration. As for how this change is propagated around the border cells, we really aren't going to be able to get at that by atomic force microscopy. We really need to cleave the membranes and watch recoil to calculate, is it only as the border cells are in contact with these nerve cells that is driving the stiffness, or is it, how fast is it propagating through the tissue? And we're working to develop tools to try to do that. But if anyone has any suggestions for how to visualize the dynamics of this stiffness change, I'd love for advice.
And I have a second question, if it's possible.
Yeah, go ahead.
It concerns the localization, yes, of the active myosin. So from the still images, it seems to be really located all around the cluster. From the dynamic images of myosin, it seems that it makes dots a bit everywhere, but I don't know how deep it goes inside the cluster itself. So is it completely, well, is it only on the surface of the cluster? And if it is the case, is there any polarization between the front and the rear of the cluster?
So what I would say is that it is largely at the interface of the border cells and their substrate, the nerve cells. And it seems to, at least, I think it's just moving around the cluster. So it's not always enriched at the protrusion. We have not looked at whether there is any enrichment. We haven't noticed, but we haven't gone through and quantified images to see if we see a striking enrichment at a particular location.
Thank you.
Right, great. I think our next question is from Lara Cavallo, coming from YouTube. She says, great talk. And do the border cells in the mutant manage to reach their final destination? And do the migration and delamination phenotypes change when knocking down Fascin with different cell type drivers?
Yeah, so we have not looked at delamination in the context of the RNAi knockdowns. It's just a challenging genetic combination, but definitely could. So we don't know who's responsible for the delamination. We could go back to our fixed imaging data because you can actually see it on the early part of the migration, an increased delay. And then they do ultimately reach their destination, but I will say, that the micropiles, so the border cells ultimately aid in the formation of the micropiles. The micropiles don't look normal, but this is a real problem because Fascin is also required for some later steps in development. So the egg is massively abnormal. So we really would need to go in there with the RNAis and really get at whether the micropile is okay. The Fascin mutants are sterile for a lot of reasons.
Thank you. Next question is from Yongo Bay. Does Fascin knockout affect focal adhesion, like FAC activation, that is also important for regulating cell stiffness during migration?
So we have not yet looked at FAC, and primarily we haven't because the border cell field really is not thinking that focal adhesions are playing a critical role, given that there is little evidence of the ECM surrounding these. But it is something with our finding on the diffuse pattern of integrin on these border cell clusters that we're interested in looking at.
All right, great. By the way, somebody had their hand raised, but I think he had to go at the hour, so I can maybe send you his email address. You guys can get in touch. And are you okay to just stay a couple more minutes to? Okay, great. So a question from Nile Tanuja. Since stiffness is dependent on both tension and viscoelastic material properties, do you envision models other than direct steric interference with myosin 2, for instance, through a general alteration of actin architecture? There's a lot of alliteration there, which could then affect myosin dynamics. Did you also characterize changes in F-actin?
Yeah, so that's a really great question. And I would say that I probably would suspect that Fascin is doing some other things to modulate myosin activity that we don't understand. I will say that we know later in oogenesis that cortical actin integrity is perturbed when we lose Fascin. But we did not notice any overt changes in the F-actin on the border cells. Although that said, we didn't look by super resolution or other means to really get at what might be changing there.
Thank you so much. I think that's the last question from Hani Suleiman, and you sort of answered that, but says, great talk. Do you think the Fascin only controls myosin activity or changes the way myosin binds to actin filaments?
Yeah, the changing the way it binds to actin filaments is an intriguing idea. I don't quite know how to get at that in our system. I think that's probably going to require some in vitro studies, but it's an intriguing possibility.
Thank you so much. I think there was one question in YouTube which we are missing. Yeah, the question is from Fanglin saying, have you tried to manipulate anything in ECM?
So because the field has largely thought that ECM isn't important, the ECM project has been my project in the lab because I don't want anyone in my lab to be fighting with the field. And because I'm stuck in my office writing grants and papers lately, I have not done any studies, but it's definitely on the docket. We have a box of flies ready to do all those experiments. So I predict the ECM does have a role. I don't know whether FAS is impacting the ECM, but I would imagine changing the overall stiffness of the environment is contributing to changes in ECM alignment and ECM stiffness.
Thank you so much. I think, unless somebody else has another question, I think we are in the end of our question answers. Jen, do you think we're missing something?
YouTube's all set. Oh, one more. There's
So
always
more.
more last minute. No, this is fantastic. Tina, your talk generated so many questions. This is really wonderful. Another from Nile saying, related to the ECM, the Scorides lab has shown ligand independent activation of integrins. Okay, so a comment, not a question.
Yeah, so I agree. We definitely need to determine if this change in integrin is due to inside out or outside in signaling. Again, it's my project, so it's moving at the speed of a snail or maybe slower than a snail.
Happens.
Not enough hours in the day. All right, well then with that, I'm gonna end the official recording. So to everyone watching,